This conference will now be recorded.
Okay. I'm not sure where I'm supposed to stand. I'm just afraid I'm going to fall off. Okay. This will work. Thanks, Felix, for that introduction, and thanks for everyone. I think the amazing thing is how much this community has grown in a short period of time. The number of registrants here is a testament to this, but also many of the people I'm sure that'll follow online or come to future meetings, and I'm looking forward to supporting this whole community as we continue to grow. Okay, so today I want to talk to you about the encoding and decoding of specificity in antibody repertoires, and you'll see why I use that analogy in a minute. But first, it's obvious that the main reason so many of us are here, I'm trying to see if I can get rid of that thing. Okay, the main reason why many of us are here is the fact that the immune repertoire, whether they be T cells or B cells, are a tremendous amount of diversity that they hold, and that through this VDJ recombination process, we have this exquisite amount of particular specificities that's coded in the repertoire. And these end up being the sources of things like diagnostics and therapeutics and all types of other aspects. So I'd like to use this as an analogy. So the communication process basically is something very simple to think about, which is when you send a message, it has to get encoded, whether this is a digital message or analog, and then this message eventually has to get decoded and received by the receiver. And in reality, this is basically what so much of what we are doing here in this community. If you take this analogy further, the sender is essentially the host or the subject that you're studying. The encoding is essentially the repertoire, the VDJ repertoire, which is the recombination, and the repertoire itself is the message. So basically the diversity, the distribution, all the various parameters. And we decode it by sequencing it, and then we receive the message through some form of analysis. Okay, so this is essentially the process that's going on. Then if you want to take this analogy further, there's things like noise related to biases and errors, and then finally we need a way to give feedback, which might be the development of therapeutics and things such as this. So this is effectively a nice analogy, and if you want to use this further, there's the, okay, thank you. There's the issue that we could basically describe immune repertoires as the language of the immune system. And that if we can basically decrypt this language, decrypt the messages buried in repertoires, we would be able to transform that into information, first and foremost, things like antigen specificity. Also, we would maybe learn about the history of the immunological exposure of a subject. We might also know the ongoing immune status and also a way to develop therapeutics. And I'll go into all of these today, and I'll just first start with the first two there. So one of the major questions, and probably one of the most fascinating questions, is, are antibody sequences shared across individuals? Like, is there, or T cell receptor sequences? And this is the concept of convergence. If convergence existed, then then there would be the possibility to transform this into potential therapeutics. One could use these for novel disease target discovery, as well as finally maybe biomarkers. So this is a really, really central question for the entire field of repertoire analysis. And you can go back very historically to this paper from Linus Pauling and Emil Zuckerkandl, and their very simple statement is that evolutionary convergence, when it occurs across different proteins basically, is essentially suggesting convergence towards similar functions, okay? So if you extrapolate that concept, because antibodies and T cell receptors are of course proteins, convergence in immune repertoires from different individuals would also represent a selection towards similar specificities. So this is a very, I think a very sound hypothesis with lots of data to support it. Now, the problem is that we always talk about the probabilities of sharing sequences. And if we decide to make a very simple analysis, no assumptions, keep it as simple as possible, we say that hypothetically, let's say diversity around 10 to the 13. I know there's a lot of debate around what that real number is for theoretical diversity, but let's keep a sort of common one that's going around, at least for antibodies. Physiological diversity, we know, is basically the number of B cells or T cells or in mice or humans. And so probability of any given sequence occurring is quite rare, 1 in 10 out of 13. The probability that that given sequence would occur in both individuals would be extremely rare. But that's not, and so that's obviously the case, but that's not how we should actually be thinking about this. Of course, the more proper way to think about this is the probability of any sequence occurring in both individuals. And if you do that, to have a 99% chance of seeing any convergence at all, you only need about 10 million cells. And that's assuming there's no bias in recombination, which of course there is. So the main point is convergence does occur. It's valid, it's statistically verified, but also experimentally verified. And if you really wanted to look at probability distributions, there's a nice tool that many of you are aware of or have used from Thierry Mora and Alexander Walczak called Igor. And that's a nice way of modeling the probabilities based on actual experimental data. Okay, and so to show you some examples of convergence, we've seen from a paper we published a couple of years ago with Victor Greif that there's a lot of overlap in the CDR3 sequences of naive B cells from mice, whether they be bulb C mice, C57 black 6, or even pet shop mice. So this is occurring quite a bit in mice. There was, of course, 2 papers that came out earlier this year that validated this as well, both from with Brian Briney and Dennis Burton and Jim Crow's group. And so we're starting to see that there's clearly a convergence here across different people. And obviously, if you took this farther and had more and more individuals, you just start to see more and more common sequences. There is some evidence as well of antigen-specific convergence, and this was work, this was actually Chudakov and Walczak who collaborated to demonstrate using a nice statistical measure using the probability distribution tool that there are antigen-associated TCR sequences in CMV and diabetes. In Jim Crow's lab as well, there was a discovery of common clonotypes in HIV among 3 different patients. And Steve Quake's group had this peanut allergy antibody that they discovered in 2 different patients, which looks like this in the CDR3 and L3. And there's a few other papers as well, and I think others that I probably have missed, but that this is sort of getting to the point that there's a convergence occurring. But these all suggest that this is more or less rare and stochastic, perhaps. Perhaps it's not, the obvious thing that's occurring all the time. So one of the important points, though, is to how do you define a clone? Because a clone is directly related. How you define this is directly related to whether you think there's convergence. And so obviously we take, cells and we sequence them, and from that we have to infer a lineage or some type of clonotype measurement. And so the really, really common ways to define a clone, and probably the simple way to do it, is to take an exact V gene and J gene and then create sort of a similarity threshold of CDR3, which many papers have done over the years. 80% is often the kind of limit of what you see in the literature. Now, the problem here is this looks at clonality from a B cell perspective. So take hypothetically here, these are 2 different germline genes from mouse, and they only differ by 2 amino acids. And if you were to look at this, these would be defined as 2 different clones, okay? Because they have 2 different germlines based on these common definitions. But the problem is that these might actually very well bind the same antigen based on obviously other factors like the CDR3 and the light chain. But then from an antibody specificity point of view, it's actually they would be the same clone. Obviously not from a B cell biology point of view, that would not be the case. So to kind of go in this direction, researchers have been, there's the traditional way of using Hamming distance, so a distance-based metric, so a threshold that you might want to put in, and some really admirable work computationally and statistically from Frederick Matson's group and Stephen Kleinstein's group with a pair of papers are trying to redefine perhaps a clonotype by using actual sequence space and not just the sort of definition based on V and J genes and thresholding. And these are really interesting, and the authors of these papers are here, so please ask them more. I'll probably ask them questions as well about it. Okay, so, but certainly the best example of redefining clonotypes is two papers which we could probably say at this point are landmark papers for our field. And this was from Paul Thomas's group, and the other one from Mark Davis and Jake Glanville. And what these papers did is they defined a clone or based on the specificity towards a particular peptide MHC antigen. So it's obviously very blurry here, but in the Thomas paper, what they are doing is essentially calculating a distance score, and they're weighting CDR residues differently, especially CDR3, they're giving more weight to in terms of defining a similarity. And the really nice outcome of this is visualized with these sequence logos. And if you take a look at this one here in particular, this is a CDR3 beta, and it's using multiple germlines and still binding the same MHC peptide epitope. So what it's really validating is that when you look at specificity towards a particular antigen, you are not reliant on a fixed germline, and you're not reliant necessarily on a fixed CDR3 threshold. You see here, this is a single germline, but a huge variance in the CDR3, far beyond any of these 80% clonotype definitions. So I think these were really important papers to help solidify this in the TCR field. So the question that we had is, does this convergent selection occur in antibody repertoires? And in this case, we're studying this in mice. And one of the questions we wanted to ask, I mean, we know that it occurs based on these previous papers that I've already mentioned, but what we really want to know is it systematic? Meaning, is it independent of antigen? Is it independent of, like, is it reproducible across many different mice? Or is it just a sort of one-off event that you find if you look deeply enough? And once, if you could identify these signatures, are they predictive? Could you actually predict what the mouse was exposed to? And could you prove that they bind to the target antigen you say it is? And the way that we decided to tackle this approach is to actually use the sequence space repertoire analysis based on deep learning. So most of you are very familiar with all of the hype around machine learning. Much of it is, of course, in the popular media, but what you may not have necessarily been exposed to is how this looks in terms of biological sequence space. And essentially, you are just, in this consideration, you're taking data sets of sequences, whether they be DNA or protein, and you're extracting features, and you're going to take those features to fit a model, and the model is supposed to tell you about an output, about some sort of basically a function of f of x fit to y. And there's lots of really common approaches here that are simple to understand, like linear regression and logistic regression. And that would be a supervised version of machine learning. There's also unsupervised, which I'm sure many of you have done, like principal component analysis and, of course, clustering. So these are tools that are out there that are available now quite ubiquitously. And in the kind of development of this field and the access to data, especially when you think about the massive influx of DNA sequencing data and computational power, deep learning has become a valuable tool. And when you encode data in sort of a neural network, what you're basically doing is taking an amino acid sequence, in this example here, and you're turning it into a matrix, and you can encode it in different ways. The simplest way to do it is called a one-hot encoding matrix, which essentially takes every amino acid, and then you plot only a 1 if it's the correct amino acid and a 0 otherwise, so it creates this sparse matrix. But it creates an input that you can put into a neural network. And really the way to think about a neural network is it's just an approximation function machine that's trying to fit the data. You're going to use a lot of what are called neurons in between here as activation functions, and you're going to train this model either with a supervised approach or unsupervised approach, and then at a certain point you're going to apply this algorithm called backpropagation or gradient descent, which tries to minimize the loss function. And the hope here is to essentially discover the weights and biases of this network, which is essentially a series of equations that allows you to map it to an output. Okay, a certain type of neural network is called a variational autoencoder. And if you look at this, it's interesting because we're using the example of hand-drawn digits. It's like the MNIST data set that many of you might have seen if you're paying attention to neural networks. And an autoencoder is interesting because its essentially goal is to construct a neural network where the input is the output. Sounds a bit strange, but it'll make more sense in a minute. But in order to create a neural network that, and in this case, it's using the grayscale values of every pixel of this hand-drawn digit. So it's a 28 by 28 matrix of pixels and the grayscale value there. And the hope there is that when it takes the input, it trains the network well enough to create the 4 again, because it has an encoding, an encoder and a decoder. So basically 2 neural networks on each side of this. And along the way, it compresses the data into the latent space. And that's really where the action happens and why it's so valuable. So if you look at this, so this is a neural network after it's been trained on digits from 0 to 9. And what it does here, it's hard to see, but basically this is the latent space of the clusters. And what you see here is that there are clusters that all map essentially to the same number. And then things like 1 and 7 end up close to each other because they kind of look similar, right? And once you look at this latent space, you would be able to pretty quickly define what numbers go together. And so this was an unsupervised approach. We didn't tell the neural network this is a 1 or this is a 0, but it ends up discovering this through this clustering approach. There are iterations of this that we have actually implemented. So there's something called variational deep embedding, or which is the final version here. And it's just an approach where you train the encoder as you are doing the clustering process. And it was developed by a Chinese group. So it's very similar to here. It's just a slight iteration of this. Okay. So now how does this apply to antibody sequencing data? So we had an experiment where we took mice, and there were 4 groups of mice immunized with 4 different antigens. These antigens go from simple to more complex, like henaglycosine, ovalbumin, RSV, and a variant of KLH called BCP. And these mice were immunized either once, twice, or 3 times, so they have varying amounts of antigen exposure. There's about 10 to 12 mice per group. And then we isolate bone marrow plasma cells from these mice, and we perform sequencing on the IgG VH chain. And we do this using Illumina 2x300. We incorporated a barcoding method that allows us to have very good error correction and bias correction in our data sets. So that's sort of basically the sequencing aspect of this. And when you first just look at a very sort of baseline analysis of clonotyping, and you have different kind of linkage functions here, but we kept the same V gene, same J gene, 80% CDRH3 similarity. What you notice is there's a lot of overlap across the antigen groups. And so what this means is it's pretty hard to know, you might be able to extract patterns from here, it's possible, but it would be, require a lot of like dissection of the data, because it is very noisy in the sense that because I suppose there's so much recombination bias in B cells that you see a lot of overlap. And this is something that we, of course, have observed previously in naive B cells. So this makes sense. Okay, so that's the problem with just using a simple clonotyping metric. But if you now create this, so now what we're going to do is we're going to take these sequences, we're going to put it into an encoder, so using one-hot encoding, and the encoder is going to compress the data into a latent space. And really the hope here is that the latent space discovers clusters that only emerge in mice that receive the same antigen. And it does not, and then it'll find a lot of other clusters too that are not exclusive to a particular antigen group. And so if you're interested, that's just basically the encoder architecture. You can see here that we feed it in with multiple layers and attention gates. Okay, so this is actually so all of the data is trained at once, unsupervised, and these t-SNE plots were generated with all of the data together, but it's only plotted for one mouse at a time here for simplicity of visualization. And what we did is it was a 5-dimensional latent space, and now we are plotting it on 2 dimensions. So just keep that in mind in a minute. But this is what the latent space looks like of 2 mice that receive 2 different antigens here in red or blue. And this doesn't necessarily, nothing automatically pops out here, but you might, if you start to look closer, notice things that are present maybe in one mouse and not the other. Just a few differences there that are important to highlight is that nonlinear interactions in the data can be captured with variational deep embedding, which is much harder or basically not possible with a simple clonotyping approach. So now if you look closer at these 2 latent spaces, you can merge them, overlay them on top of one another, and zoom in. And anecdotally, if you wanted to just visually look at it, you could see that in red here is the RSV and in blue is the ovalbumin. And you might say, well, aren't those 2 different clusters there? They look like 2 different clusters in 2-dimensional space, but in 5-dimensional space, they would essentially be very close to each other. You can extract the sequence logos from these clusters, and they look something like this. So it's not like it's exotic away from a clonotyping metric. You can see by eye some similarities and patterns, but you can probably also see that things like 80% threshold would not hold here for most of these sequences. There's no doubt that these sequences are using different V genes because of the CDR2 diversity. So it's capturing some effects here that aren't normally possible. Okay, so now the question is, can we take this latent space information and identify patterns that are predictive for antigen exposure? So are there clusters that occur only in mice that received a certain antigen and not in mice that received a different antigen? Okay, so we zoom in again, and we essentially can identify the clusters that have, through a permutation, non-parametric permutation test, but we can essentially identify the clusters that are emerging in mice that receive the same antigen and not in different antigens. So these are broken up by the antigen groups, and each column is a different mouse, and each row is a different cluster. And what you see here is that, let's just look here for easy purposes, that the RSV has these clusters present in, not every mouse of RSV, but a lot of them, and then they're almost never used in mice that were receiving a different antigen. You can zoom in again, and this is actually what they look like across these mice. So the previous logos I showed you was just within one mouse, and now this is across multiple mice. And then you can again look at the latent space here. These are OVA plots of each individual mouse. And the important thing, if we were saying, and just zooming in on here, we just see that not every mouse will use that cluster. So it's not something that is going to be in every single case. Okay. So in order to test to see if these clusters are predictive of antigen exposure, we just did an SVM, and we first randomized the labels. And you can see when you randomize the labels and do a 1 versus all support vector machine model, you can't predict antigen exposure based on these clusters. That makes sense. But once you actually assign the correct labels, the prediction level is extremely accurate. So essentially, these clusters are representing the antigen exposure quite well. Think about this maybe as a digital ELISA, as a way to perhaps extract information that way. All right, so there's still a major question, which is, are these binding the antigen? And this was something that we spent an enormous amount of time and energy on, which will be condensed down to just 1 or 2 slides. And what we did is we had heavy chain sequences from these mice, and we did not do this in a paired fashion, single cell fashion, so we did not have a light chain. So we basically took the heavy chain of some of these clusters, the ones that were convergent, we synthesized them, or synthesized them for us, and then we extracted RNA from the light chain of these mice. We extracted RNA from these mice and amplified the light chain library. So one mouse per each antigen group. So basically the light chain libraries is only coming from one mouse per each antigen group. It's not mouse matched. And then we tested that light chain library with the heavy chains. And for each antigen group, just in bulk, you can see that we produced these convergent clusters are binding the target antigen. They don't appear because all these additional lines here are binding to the other antigens. So they aren't just like polyspecific sticky antibodies. They are indeed binding to the correct antigen. And then this was a mammalian surface display system that we developed. So it allows us to also isolate single cells by FACS. And we can show you that for 7 out of the 8 clusters, convergence clusters we tested for ova-albumin are antigen specific. 4 of the 8 we tested for RSV were antigen specific. So we definitely believe that these are antigen-specific binders. And a really important thing is that some of these, so each one of these dots represents a given cluster in each mouse for each antigen group. And what you're seeing here is the highest ranked variant based on abundance. And the main point of this is there are variants in a given mouse that the highest ranking in that mouse is over 1,000. But yet it's still part of this convergent and antigen-specific group. So it means we're finding things buried potentially deep in the repertoire and not simply just things from clonal expansion alone. Okay, now within a given convergent cluster, this is for this RSV grouping, we decided let's take we only had one variant that we used for the previous binding data. So now we said, let's take additional variants. So we synthesized 15 variants of this based on this logo that were from the 10 other mice. So these are all variants coming from a different mouse. And you can see here, if you were just to align the CDR3, that a few of them would pass the 80% threshold, but there's about 10 of the 15 that actually don't, that are more than 80% away. So they would not have been classified as a clonotype. And some of them in these mice are ranked very low in terms of they're not highly abundant sequences, so they're buried in the repertoire. And then when we tested them for antigen specificity, 15 of the 16 that we tested were antigen specific. So indeed, these convergent clones are antigen specific, not just a representative clone, but the variants of them, at least a large fraction of the variants appear to also be antigen specific. And they're using light chains, remember, and we fixed the light chain here, so it's not even the natural light chain, and we can still recover this binding. Okay, another really interesting thing, going back to the paper that I mentioned earlier from Paul Thomas about TCR distance, we decided let's look a few of these clusters a little bit more carefully. So this is the ones I'm highlighting. And what you can see here is that in some cases, like with ova-albumin, this cluster, convergent clone, you see there are many V genes associated with it. Now it's not clear that every single one of those V genes combinations here are actually going to be antigen specific, but it's very possible that several of them are. And you can see this trend go down. And so what this is showing you again is the specificity towards antigen is not fixed to a particular germline, and shouldn't be fixed to a CDR3 threshold also. So there's really a lot of diversity here. Okay, the last point I wanted to make on this story is that I talked a lot about this part here, but I haven't mentioned the decoder. So the decoder essentially takes the latent space, and remember, it's trying to reconstruct it. It's trying to reconstruct it into essentially the same, if you want to think about it that way. And that's just the decoder architecture if you're interested. So what do we use the decoder for? Well, decoders is a form of generative modeling, and this has become very popular in the press for many reasons. But one example is that generative modeling has been used for is to essentially take pictures of real faces and then take some random noise and build a neural network. And sometimes you use something like a generative adversarial network is one example of this, although autoencoders can also be. And you want the generator to essentially create novel variations here, essentially novel faces. And so this is a really cool website. These are essentially faces created by these generative modeling. And so these are fake faces. They don't exist. And you can see how well they are at essentially recapitulating the features of a real human face. So how does this look for sequencing? Well, on the sequencing data set, what essentially we want to do is go into the latent space and ask the decoder to create sequences that don't exist naturally, but look as similar as possible. And what we'll end up discovering is combinations that weren't existing in the biological repertoire. Some of these combinations, if you look here, so the red would be real biological sequences, the blue would be the generated sequences, and they'll end up covering different places of the repertoire or sequence space. And you might say, what's the point of that besides just, making more kind of novel sequences? It's the possibility that some of these generative modeling sequences, many of them hopefully are antigen specific, but the ones that are antigen specific have unique features that make them better perhaps as a therapeutic antibody. So things that improve their biophysical properties. So that would be the engineering concept here. And we're still in the process of actually selecting these sequences, expressing them, and testing these parameters. Okay. Good. All right, so I hope I've shown you a little bit about antigen specificity and immune history. Now I want to talk about immune status. So there's something that we all do from our data sets is that we get a lot of information, and what the hope of this field I think has in a lot of ways is to be able to eventually use this as a diagnostic metric to tell you whether someone the repertoire composition, whether it be TCR or BCR, tells you whether a person is healthy, diseased, maybe it's an early prediction of prognosis, and that this could be done using the repertoire composition in of itself. And so this is the hope here. And there's some simple examples like this, conceptually simple, like lymphoma or minimal residual disease, where there's diagnostics that use just clonal expansion as a way to tell you whether somebody is potentially undergoing some type of hematological malignancy. But what we do know is that there's a lot of additional information besides clonal expansion. And so this is a collaboration with Victor Greif at University of Oslo and Cedric Weber, a PhD student in my group. And the hope here is to create, to use all of the important information that comes from repertoire sequencing. So not just clonal expansion, but also the germline diversity, a sequence similarity network, amino acid usages at positions, also some occurrences of patterns, including potentially immunosignatures. So as we learn antigen-specific motifs as well, we could embed those into this multi-layer analysis. And basically, it's a multi-layer network. So you take all of these different features and you try to effectively understand how it looks in one composite network. And the hope is that people that have certain immunological phenotypes, certain types of disease, will cluster together once you use all of this information. And this would allow us to then extract a person's health. So what we've mostly done at this point is trying to look at various features. So the ones I mentioned already are pictured here, and you can see some of the details here. And I should mention that Cedric has a poster at the conference if you want to talk to him about it in more detail. And so we did this both with natural data sets, but also some synthetic generated data sets. And so the hope is to see if, do these features, first of all, are they relevant, and are they overlapping? Are they submitting us just generating redundant information? Otherwise, they're not adding that much to the whole process. So this just shows you basically that the orthogonality and sufficiency of the features is quite complementary. So there's not much mutual information coming from them. So it seems like we've settled on ones that are useful and worthwhile implementing. And then this is data sets looking at the sensitivity analysis, and it's a little bit difficult to see here, but we have various compositions here. So things at this point, the overall structure of this immune net approach is well benchmarked. And our next goal is to essentially use this to decode the immune status of human samples. And this would be a large-scale approach to look at many different human immune repertoires. We've started collaborating with Zhao Lu from the BGI Shenzhen to look at this across many different types of human individual patients. And then hopefully at some point, if we might also start to use machine learning to help us to classify this. But this is an ongoing project, and we're excited to see where it leads us. And so just for a couple of slides also, I want to talk about physiological convergence. So the thing is that the immune system has a physiology to it. It has not just this floating around cells, but they're embedded within structures and organs like lymph nodes and spleens. And kind of a central question is, what is the overlap of these immune repertoires, especially the overlap as how it pertains to specificity? And so there was a really nice paper from Nina Prack's group a couple of years ago that looked at this with human samples and really started to identify the sort of physiological network of clonal lineages that exist. And what we wanted to do is to build on that work and to address this in the context of specificity. And we did this in a mouse model, and the reason why is that we could control specificity, obviously, just by immunizing the mice. And we had two different sets of groups, or actually 4 different groups. I'll just highlight 2 of them, which is a subcutaneous immunization, either a single immunization or multiple immunizations. We also did a IP injection. And we have a lot of organs and a lot of sequencing. So what I'm showing you today is just a partial representation of the data. And Lucia has a poster at the conference as well, so you can talk to her about it. If it gets printed, hopefully. Okay, so these are sequence similarity networks of clonotypes from, so this is not an advanced clustering here, this is just very simple clustering for, but I think the phenomena is so clear we don't need anything so advanced at this stage. And so these are mice that received a single immunization, and what you can see is that these are each of the different organs, the lymph nodes on top, bone marrow and spleen here, and they're color-coded, which I'm sorry you can't see very well, but color-coded to see if it's present, if that particular clonotype is present in a different organ. And what you see is a single immunization creates kind of a sparse, if you will, network here. But after multiple immunizations, what you can see is there's this drive towards convergence across the organs. And our very simple hypothesis, which we will have to validate in the coming months, is that as you drive this physiological convergence, that sequences that are present across multiple organs are actually the antigen-specific sequences. It's probably intuitive, and hopefully when we do it, it'll work. But nevertheless, I think it's something that has not quite fully been validated, and it's really exciting to see that we can control this physiological convergence, and then we can show this is antigen specificity. And I think this again comes back down to immune status, because titers alone don't tell the story. And this is, I think, telling us a lot more about physiological diversity. Okay, and in the last couple of minutes, I just want to talk about slightly different iteration of this, but how we can use immune repertoires and the idea of sequencing to build better therapeutics. Okay, so everyone is fully aware that monoclonal antibodies are a really powerful way for building, therapeutics against many, many different diseases. And so the issue, though, is that you have to understand and appreciate that a monoclonal antibody that binds target antigen is not a drug. It's far from being a drug, because what makes a drug requires a massive amount of other parameters to be satisfied. So if you find an scFv from a phage library or a yeast library or even a hybridoma clone, even if it's a human antibody, it's very, very far away from being a therapeutic candidate, because it needs to, in addition to having the right affinity and biological function, it needs to have the right PK/PD parameters, it has to have a reduced immunogenicity, and it has to be the right developability parameters, including, lots of things around solubility and stability. So to really highlight this, there's a company that leads, is one of the best in the world at generating antibodies for therapeutic purposes, and it's called Adimab, and it's from a spinoff of Dane Wittrop's group at MIT. And they did this really extensive study where they looked at many different antibodies, all either in late-stage clinical development are already clinically approved, and they showed that they have very different developability parameters. And in some cases, the developability scores were predictive to essentially side effects or problems that patients have. One of the best examples here is these are antibodies discovered from mouse hybridomas, or hybridomas in general, and these are antibodies discovered from phage display. And they called something that had a low developability score flag, and you can see that phage antibodies have many flags, meaning that they're not optimized for the right parameters. And this might be one of the main reasons why, despite having, phage display, of course, winning a Nobel Prize last year and having been around for quite a while, has very limited success clinically. There's only about 4 or 5 of the 80 antibodies clinically approved came from phage. And a lot of reasons why is because these developability parameters are very difficult to optimize. So what we wanted to do is to try to address this problem, this bottleneck of take a binder and make it a drug. And this is really, one of the most important problems that, at least in pharmaceutical drug development for antibodies, that exist. And so this study was done by Derek Mason. He's not at the conference, but we have the publication online now on bioRxiv. And what we did is we started with a therapeutic antibody, Herceptin, which is against HER2 breast cancer, one of the highest selling drugs in the world. And we mutated its CDR3 domain using a mutagenesis strategy based on deep mutational scanning. This mutagenesis strategy allows us to identify positions in Herceptin CDR3 that are predictive, or not predictive, sorry, that are essential for specificity. For example, this tyrosine Y105 residue cannot be mutated. If you mutate that, then you lose all specificity. But many other positions are enabled for mutations to maintain specificity. So we used this deep mutational scanning data to build a combinatorial library, and we screen a very small combinatorial library, again using our mammalian display system. And what comes out of this combinatorial library is about 10,000 unique variants that bind HER2, and about 30,000 that don't bind. So we have like basically this forms training and test data. So we deep sequence those positives, and we use it to train a neural network. And the whole goal here is that the neural network should understand, or should be able to extract patterns buried in this combinatorial sequence space that predict antigen specificity. And then after training, so the theoretical diversity here of the combinatorial library is about close to 10 to the 9. Okay, that's far greater than you could ever screen in mammalian cells. The best pharmaceutical companies like Genentech will only screen around 1,000 IgGs per development campaign. So you could never screen that entire space. And we screen, because of our mammalian display, we actually screen more than most of the companies do with about 30,000 variants, 40,000 variants. But what we wanted to see is if we now use the neural network we've trained, and then we put in an in-silico set of sequences, basically the other 10 to the 9 minus 40,000 that we didn't train the model on, can it predict which ones are antigen positive? And then once it predicts which ones are antigen positive, we can run them through all these developability measurements, not measurements, but actually metrics that you can do in silico. And then at the very end, you have a small set of sequences that you can test for the proper pharmaceutical properties. So we use 2 different types of neural networks, which are very popular and used across all of machine learning, but they're quite good as well for biological sequence data. So it's a long short-term memory recurrent neural network, LSTM-RNN, and a convolutional neural network. So they're both being trained, right, with our 10,000 positives and our 30,000 negatives. And when you shuffle the labels, these networks aren't able to predict anything, which is good because that's what you want. This is a ROC curve. But when you have the correct labels, you can see that they're achieving extremely good true positive to false positive ratios. The area under the curve here is almost 90% for both models. So it seems like even though our neural network is trained on about 50,000 sequences, and the theoretical sequence space is 10 to the 9, it's doing an extremely good job. And I should have mentioned, of course, we have separated the training data from the test data. So there are sequences here where we have ground truth knowledge of binding or not binding, and that's what these metrics are based off of that. Okay, now we took to validate, we now took 30 random sequences that are 5 amino acids away. You can't see that very well, but they're at least 5 amino acids up to 8 amino acids away from the 10 amino acid CDR3 of Herceptin. So we've completely, in many cases, recoded the CDR3 sequence, and we recombinantly expressed these 30 sequences. They were not in the training data or the test data, and all 30 of them were antigen specific. So essentially the model, and this was using a filter of higher probability score from both models, of which 3 million sequences satisfy that criteria, and all 30 for 30 were antigen specific. Most of them were sub-nanomolar, and including even one that has a better affinity than the clinically used Herceptin. So this approach is extremely good at identifying antigen specific sequences, but then the really great thing is we could run our 3 million sequences through many developability filters. The experimental sequences, which is about 11,000, which is tenfold more than even possible with industry methods, will get us about 17 sequences that pass all the criteria, but our deep learning predicted variants give us almost 9,000 variants that have passed that filter. And then when you plot the developability score, you can see, you could find some good experimental variants here, but the deep learning predicted binders have even higher developability scores. In fact, there's close to 500 sequences that have better scores than what we would have found experimentally. And then remember that not every sequence has very high affinity. So these 17 experimental variants, many of them may not have the affinity that you need, but we could be very confident that within these hundreds or thousands of deep learning predicted variants, there's ones that have the exact specificity or the exact affinity that you need. Okay, so with that, I just want to summarize because Felix has moved into the front row for some reason. So that I think, and we should have highlighted well that basically convergent selection appears to occur systematically, at least in mice, and that they are really predictive of antigen exposure, but also antigen specificity. And using the tools that we have, I think we can extract these features, and there will be others, I'm sure, in the future as well. And many people are working on different approaches to this, but one, I think, real powerful way was using variational deep embedding. I think ImmuneNet is a really exciting way to look at clustering of immune repertoires, so not single sequences, but entire repertoires. And now we are testing this out on real samples. The physiological convergence of antibody repertoires is influenced by immunization. It's influenced by the exposure to antigen. And I believe eventually we'll be able to validate that it's overlaid with antigen specificity. And finally, I think deep learning is a really powerful way to do a multi-parameter optimization of therapeutics. So with that, I thank everyone in my group. I've mentioned a lot of people along the way and collaborators as well. So without taking up any more time, I'll be happy to take questions.
Great, well, that was incredibly exciting, and I'm guessing there will be questions, and then we'll also have some time, during the days to talk to Sai, but let's open it up to questions. I'm hoping he can answer, and we can use this in the audience.
Hey, Sai, really nice talk. Thank you so much. Yes, so in that last bit that you were presenting about, that was really exciting. And I was wondering, my memory might be wrong, but I'm thinking that Dev Sidhu's libraries that he's built, which are phage display, but they're built around a particular framework, and I think it's
actually Herceptin.
Yeah, exactly.
Yeah.
It's a Herceptin framework. And there he's put in variability at the CDR3s, which I think is a little bit like what you're doing in silico. And I was wondering if you guys have compared notes on some of these antigens. That's the first thing I was wondering. And then secondly, I wanted to ask you about, you've been talking about antigens, but not epitopes. And so I wanted to see if you can see in silico or identify antibodies that may be interacting with the same core of critical binding residues. and yet have very different sequences. So could you pick out things that may be competitive with each other for an epitope?
Yeah, okay, so first with the libraries that DevSidu has built, yeah, you're correct, they're built on Herceptin and they have a minimal diversity in CDRs. We haven't really talked in detail about applying these tools to it, but more just looking for different types of clustering of it. And I think it's an interesting way, and what should be pointed out is nothing that I presented here is relegated to just mammalian cells. It could be used for any directed evolution experiment as long as you have a good selection criteria, like say binding target, and also if you have, of course, sequencing as well. So the other point is actually goes right into that. So epitope specificity. So I don't think a model, any type of machine learning model will work if you're changing epitopes, because your training data is really optimizing a very small solution space around binding a particular target epitope. So in the case here, we've mutagenized the CDR3, but if our training data was built on essentially 40,000 variants and close to 10% or so of those are antigen binding, it means they're really binding the same epitope. When have you ever created an antibody library before and had 10% of it binding, right? You would not have that much binding the same epitope, or you would not have 10% binding different epitopes. I can't imagine you could create an antibody library that effectively. You're looking at funny faces at me, Jamie, so we'll have to talk about this afterwards. But okay, so no, but we don't, but what we don't know yet is how to predict epitopes. I think that's a much harder question, and probably at some point might require structural modeling to be somehow incorporated.
Moved up to the new section. Good job.
So looking at all the different developability criteria, when you sample a lot of unobserved sequences, and then you look at the distribution of, solubility or stability or that kind of thing, and then you compare that to the distribution of just like completely random antibodies from some phage display library, what do those distributions look like? Is when you're sampling from, a particular antigen space, is everything like, is the distribution actually squashed that you don't really get a lot of variability, or is it actually like a huge variability and you can really select like the top 0.001% of antibodies?
Yeah, great question. And it was something before we did the experiment we didn't know what we would find. And in fact, I think it depends on which criteria you're using, but you could find extremely wide distributions. So basically, here was an example where I showed you that sequences that had a better developability score based on the metrics than the Herceptin, the clinically used Herceptin, and we still had 9,000 variants that were better. Of the full 3 million, then there's a ton that are actually worse than Herceptin, obviously, because Herceptin is a clinically used antibody. So I think that there's a pretty wide distribution. It's hard to know, it's hard to know how that would look to a completely random library, but I think it would be that very small differences can actually make a big difference for a therapeutic antibody. Not binding, and not necessarily even affinity, but there are popular cases like a metamune antibody that was stopped in clinical development because 2 amino acids made it immunogenic or had, I think, aggregation problems. So there could be really, really subtle differences that make a big difference for therapeutic purposes.